Our next speaker is Dr. Indumathi Mariappan. So Dr. Indumathi is a research scientist at the Center for Ocular Regeneration, LV Prasad Eye Institute, which is like, in the same city. She received her PhD from the Center for Cellular and Molecular Biology, that is CCMB Hyderabad. Her research focuses on basic and translational research efforts towards addressing the problems of retinal and corneal diseases using the different sources of adult and pluripotent stem cells. In 2016, she was awarded the B.M. Birla Science Prize for Biology for her contributions in the area of stem cell research. Her talk today will focus on retinal and corneal organoids for in vitro modeling and regenerative applications. So Dr. Indumathi, over to you.
Okay. A very good morning, everyone. And on the outset, let me first thanks Abhijit for the introduction. And thanks to the organizers, specifically Surat, Dr. Radha, and Madhu for roping me in to give us an opportunity to share some of our experience with respect to generating organoids, eye organoids, both retina and corneal organoids today with you. So as I've shown here, today we'll be looking at two different organoids that we have been developing in the lab and have some experiences. And the retina, it's the back side of the eye, and you have cornea in the front part, and you have lens. So all these three organs, cornea, lens, and retina, play a very important role in light perception, and then how our brain converts these light signals, electrical signals coming from the retina into visual outputs. So as you can see here, both these tissues are very complex tissues and made of multiple cell types. Here, cornea, you have a stratified squamous epithelium on the top. You have a very thick layer of stroma and a layer of endothelium on the inside, together contributing to a complex structure, which is totally transparent, as opposed to retina, which is a neuroretinal, which is very similar to your brain, and it has at least 6 different types of neurons and a glial cell type contributing to the neuroretinal part, and a single epithelial cell type called the retinal pigmented epithelium together function as a signal unit and helps in light perception. So coming to the retina, so as I said, it is a very complex tissue. You have 3 different cell layers. Outermost layer is your rods and cones. All your photoreceptors, we know that is light sensing cells, are organized in the outermost layer, and you have interneurons which transmit these signals coming from the photoreceptors towards the brain. And there are bipolars, horizontal amacrine cells, and the Mueller glia together forms the inner nuclear layer. And finally, your retinal ganglion cells whose axons reach the brain and transmit the signal. And you have another type, which is an epithelium, which is a pigmented cells, and it helps in nourishing the photoreceptors because all your vasculatures stop at the inner nuclear layer. Your outer nuclear layer is totally avascular. So therefore, these cells need nourishment, and that is taken care of by RPE. There are a lot of genetic disorders. So this is the assembly. So your photoreceptors totally depend on the RPE for its nourishment. And there are a lot of genetic conditions. This is a close-up view, how the RPE and the photoreceptors intersect in the tissue. So the photoreceptors keep shedding some of these outer segments all the time, just like how we shed our skin cells, and these have to be cleared from here. And the RPE cells does a phagocytic function and also participate in the nutrient solute transport. And there are also visual cycle intermediates that are recycled between both cell types, and there is a recycling process that is very important for the visual function. So all of this is taken care of by both the cell types. If there is any genetic defect affecting either the photoreceptors or RPE cells, then you have problem in vision. So looking at the condition age-related macular disease, as I said, it's age-related, or in case of macular dystrophies, the central part of the retina is affected. So here, a human retina is not uniformly distributed. The rods and cones are not uniformly distributed across. The cones are highly concentrated in this region called macula, which is just a 2 mm region, which is important for our bright vision. daylight vision and color vision. Whereas the periphery is enriched with rods which are important for your dim light vision, and therefore night blindness happens if there are photoreceptor rod deficiency. So in this case, because the macula degenerates, people have problems in bright light vision as well color vision. So you have central vision loss and the periphery remains abnormal. Whereas in case of retinitis pigmentosa, which is again a series of genetic disorders, a lot of mutation happens. You have the peripheral cells that are lying, rods are dying, RP cells die, and therefore the patient will have your tunnel vision. Central vision is retained where periphery is gone. So in either case, gradually the patient lose the complete retinal photoreceptors and go to total blindness, and they become legally blind. So as I said, it is both can be age related or inherited. Over 250 different genes have been so far listed to be responsible for this condition, and no treatment as of now available. Only these patients have rehabilitation possible for these conditions. And now latest technologies, gene therapy has been looked at in case of gene mutations where it is known, provided the cells are still functional, still live but not functional in the patient. So the idea is to rescue the cellular function in the residual survival cells. But if the patient has lost the cells already, then gene therapy is not the option for the patient. Therefore, cell therapy is looked at. And here the idea is to replace the lost cells and to rescue the tissue function. However, unlike your skin, or also in my subsequent talk, I'll talk about cornea, there are stem cells in adult tissue, whereas in human adult retina, there are no stem cells. So therefore, the only option is to look for alternative sources like pluripotent ES and iPSCs. Several groups around the world are looking at these options for developing retinal tissues. So that brings us to the iPSC research. So we are interested in, my lab is mainly focused on understanding retinal development, disease, and therapeutics. So therefore, our lab has been working extensively on pluripotent stem cells, and both deriving iPSCs from normal individuals, also from individuals who has documented gene mutations to derive patient-specific iPSCs. So a lot of protocols are being reported for stepwise differentiation, because basically it follows, in vitro differentiation follows the embryonic development, wherein the iPSC is undifferentiated, iPSCs mimic the blastocyst stage, and then when it differentiates, you have to push them to a neuroectodermal commitment, and neuroectodermal to an eye field commitment, followed by within the eye field you have RP, retinal pigmented epithelium, versus the neuroretinal commitment. So different growth factors have been known from the understanding of human retinal development being important for all of these steps, and this can be recapitulated in vitro using different cocktails of growth factors at different stages in differentiation. So every protocol might vary slightly in this schematic, but roughly this is the broad schematic people follow. So in the lab, when the eye field develops, you can find them morphologically, even in a phase contrast microscope, clearly how these structures look like. So these are like donut-shaped structures. These are like eye cuffs that develop within the field. So, and you have your ocular surface ectoderm, which constitutes your lens, cornea, everything starts coming out as an outer layer. So this kind of structures, you can easily find about 30 of them even in a single 6-well plate. And you can actually pick them, the neuroretinal island from the center, and you can either grow as uniform monolayers, or you can put it in suspension to develop 3D retinal organoids. And this is lens structures. You can clearly see from the morphology, from the highly translucent structures. Now we are pretty much familiar with how they look and which structures are there, and can easily pick them manually to enrich these structures. So for RP, it's pretty simple, easier because it's a monolayer. It is pigmented. I can see pigmented patches, isolate them. You have cell surface markers. You can fax sort the progenitors and make enriched population. So this is pretty much simple. We generate monolayers like this, enriched population of RP cells, and there are clinical trials already initiated for iPSC-derived RP cells for AMD treatment. So you can either make them as a sheet, as shown here on collagen matrix or PLGA matrix. Several of them people are exploring. Or you can also prepare them as cell suspension, inject them subretinally. So this, either of this can be done. So it is delivered subretinally where the photoreceptors are still intact, RPs are degenerating, and your idea is to replace the RB cells so that photoreceptors will start functioning. So same thing for neuroretina, you can differentiate these clusters, continue them in adherent cultures, you will start seeing all these neuronal structures including photoreceptors, precursors, several of them come up. The biggest challenge in 2D culture for neuroretina is their purification. The moment you detach them and they are mature cells, when you detach and then go for sorting, then there is massive cell death, and these cells don't survive, and they don't self-organize also properly as multi-layered structures which you normally see. So that's a problem with the 2D system. We struggled a lot with this and then decided to go for a 3D system. So you can pick these neuroretinal clusters at this stage before the cells migrate out, because these are progenitors, they're assembled to form these structures. When you pick and put them in suspension, they nicely form these cup-like structures, which is typically donut-shaped structures, and there are multi-layered progenitors nicely arranged. You can see the beautiful striations of these cells. So at this stage, they are progenitors. They can make photoreceptors, they can make RGCs, any of them, depending on the culture condition that you're going to put it. So when you put in addition, you can see the RGCs send out large axons. And alternatively, without even going for adherent cultures, right from EBs, if you put continue them in suspension cultures, like what Dr. Radha was showing, bilateral optic cups coming up from a single organoid. This is a brain which is CNS, or the neuroectoderm organoid. You are seeing probably this is a CNS cluster, and you see two neuroretinal clusters, optic cups coming up. Here the reporter is an RX-specific progenitor promoter-driven GFP. Beautifully, they form the vesicles, invaginate to form the cup, and the neuroretina and RP distinctions can also be made at this structure. So you can literally pick or chop these structures and then go for retinal organoid culture or whatever cell type that you want to isolate. So looking at the self-organization, I said retina is a complex tissue. Different retinal neurons constitute the total retinal tissue, and photoreceptors are on the outermost layer. You can see similar self-organization in an organoid. Here is the BRN, which is an RGC marker, and photoreceptor rhodopsin, beautifully arranged in a proper polarized manner. Though the lamination is not fully complete, but then at least the fetal retinal development stage you can achieve with organoids at 2 to 3 months in culture. And there is also important that the photoreceptors are a unique cell type, which is not just a circular cell, but they have a unique extension. Their cilia is primary cilia is actually modified into outer segments wherein the photopigments reside. So this morphological maturation is very hard to achieve in dish, but it's not impossible, and people have shown that it is feasible to generate mature photoreceptors with rudimentary outer segments. So you can see the nice laminated arrangement of this progenitors here, and you can see that here it's a CHX10 is the precursor marker. Almost every cell layer expresses at the early stage, and when it matures, you can see the outermost layer expressing rhodopsins. Its recoverin is a photoreceptor precursor marker, and they also form this morphologically mature inner segment like extensions start developing in these cell types, and if you stain for rhodopsin or conopsins, then they will be positive for the expression. So this is now possible, and if you open this is size, if you look at it, it's about 2 mm. If you open up, you will get about 6 to 7 mm linear, multi-layered complex retinal tissue, which can then be injected into the subretinal space, either as an intact tissue, or if you are interested only in injecting photoreceptors, you can harvest the photoreceptor cells here, or at a very early stage before the morphological maturation happens as precursors, and then that can be injected. So these are the options that are in front of us now for further exploration. Now moving on to cornea. So cornea, again, as I said, it is a complex tissue, multi-layered epithelium, thick stroma, beautiful collagen fibrils, and then endothelial layer. So these stromal cells, these are from neural crest-derived structures. This is from the ocular surface ectoderm. So different lineage cells coming together to form a complex organ. So what has been done so far in the clinic is, so you have the cornea, which is a transparent structure, and the epithelial stem cells reside in the periphery around about 2 mm around the margin. 360-degree cornea is where the limbal stem cells for the limbal corneal epithelium resides. We call this region as a limbus, and the epithelium as limbal stem cells. And these cells play a very important role in corneal homeostasis. Every time you blink, there are a lot of cells that are shedding, and these cells regenerate the lost epithelium. But if there is a drastic defect, in the sense like there is a burn injury, alkali injury, wherein there is not just the surface but also the limbal region is damaged, then this normal homeostasis doesn't happen. The conjunctiva comes over, and along with the conjunctiva, vasculatures also come over, and that leads to a big problem. The patient cannot see, there will be a lot of irritation, watering, and there's a big problem. We call this condition as limbal stem cell deficiency. So what is now available as a treatment for patients for this condition is autologous stem cell transplantation, because if one eye is affected, you can always harvest the stem cell from the other eye, because we know that there are stem cells and that's sitting in the limbal region. We take a small biopsy of 2 mm and grow them on amniotic membrane, which is used as a graft here. Instead of amniotic membrane, now you have different scaffolds people are exploring and using as scaffold for this culture. Can make sheets of epithelium like skin grafts. You have corneal epithelial grafts that can go on top of the patient. So that is available in the clinic. Now it's been offered as a treatment. And this is how the patient eye looks like, and once the pannus, we call this as an abnormal tissue as a pannus, which is removed, and you have the limbal stem cell graft transplanted onto the patient eye. And at LVPEI, this has been carried out since 2002, and so far this procedure has been used for the treatment of over close to 800 patients, and the success rate is about 76%. So while this is possible for unilateral cases, bilateral cases, say if both eyes are affected, then you don't have an autologous source, then you have to depend on allogenic source, or eye bank derived tissues can serve as a source. Or now you have with iPSCs an autologous iPSC derived. You can take skin cells from the patient, instead make iPSCs, and now we have a method of making corneal epithelium. It's possible to make autologous graft for these bilateral cases. So while we were looking at the retinal organoid development, we also came across the corneal organoids. So depending on the stage you pick these high field clusters and put them in suspension, you can either get only retinal progenitors or the retinal calves, or the entire eye field like clusters comes up. You can see that these cells are self-organized. This is a corneal organoid. This is a retinal organoid. Right opposed to each other, front you have this, and at the right opposing end you have the corneal organoid. You have the lens structure coming very close to the corneal organoids. So these can be dissected out. You can see there it is a very transparent, very thin tissue which is fluid filled. So if you fix them, they just collapse at the early stages. Okay, so which later on with the migration of other neural crest cells, they get thickened up and the stroma develops, and you get a thick stromal corneal organoid. Here is an example of a bilateral organoid with bilateral retinal cups. And this is how the early corneal organoid looks like. It collapses, the structure collapses, but it's a monolayer or bilayered structure to begin with. And then the stroma develops, and you can also have complex, well-developed corneal organoid, which typically mimics how a normal cornea look like. You have a central cornea, and you have a limbal region, which is like crypt-like structure where the stem cell resides, which is followed by contiguous epithelium, which is nothing but the conjunctiva, which also has goblet cells to produce mucins, which are important for your tear homeostasis. So all of these complex structures develop. They are self-organized structures, as Dr. Radha was telling, because these cells were differentiating simultaneously in the dish from a multipotent progenitor. You get all these cells coming together and self-assemble to form these complex structures. Whereas in an organ-in-a-chip model, you actually see these cells in different layers to prepare a complex structures, whereas in self-organized structures, the cells know where to go and they self-assemble. So if you look at the size of these organoids, it's about 2 mm again. So not directly good enough to substitute as a corneal graft. So you still depend on donor corneas as a corneal graft because these are miniature structures about 1/6 the size of a normal cornea. But then this can provide you ample source for harvesting limbal stem cells for any downstream application, be it for basic research or for translational applications. So they do express all the markers, like as I've listed PAC6, keratins, specific cornea-specific keratin, and the stem cell marker doesn't express skin-specific keratins. So clearly suggesting that they are fully mature organoids. So what we did went ahead. So we have been already offering autologous limbal stem cell grafts on amniotic membrane, which is feasible, nicely grow as a sheet. And this sheet goes to the clinic. We asked the question, can we make a similar sheet using corneal organoids that we have made from iPSCs? So we took a biopsies and then made these structures as sheets. And it's now possible that these cells can be used for basic applications or for clinical or preclinical studies. So what can we do with these organoids, be it retinal or corneal organoids? Now we can understand developmental biology, several questions related to biology. And for disease modeling, in case of genetic diseases, if you have patient-specific cells with mutations, you can model them, you can create patient cells, or you can use CRISPRs to edit these genes and see what happens, how it affects their tissue development. And also for testing different drugs for therapeutic applications and for even cell therapy for regenerative applications, and now even for gene therapy, because gene therapy products have to be validated in animal models, suitable animal models. Now if you have patient-specific cells and you can show the reversal of disease phenotype, then it will be a good proof of concept, in vitro proof of concept that can be used for taking the gene therapy product to the clinic. So here is an example that I put up. So for retinal organoids, when used for understanding asking some basic simple questions, here it's an example. I'm showing two different AAV vectors. Which one is good for transducing retina? So here you can see that AAV2 serotype is much better. It can infect the cells, whereas AAV8 is really worse. It's very bad. And similarly, which promoter, if you are targeting a gene, delivering a gene, you will be using a tissue-specific promoters to drive gene expression only in the cell type of interest, but not in others. Here they're examining CRX, which is a photoreceptor-specific gene, and its promoter drives expression only in photoreceptors, as opposed to a CMP promoter, which is ubiquitous. You can see pretty much the entire organoid lights up. So this kind of proof of concept studies can be done using this organoid model. Here is another example of how a gene therapy vector validation can be done. So here is an AAV CRX gene therapy vector. They're validating using a CRX null model. These organoids do develop normally. In spite of CRX not being there, it develops normally. Your retinal precursors are expressed properly, no CRX here. But then it doesn't mature because it's important for maturation. So none of the mature markers like opsins express here. But when you treat these organoids with AAV CRX vector, these organoids go on to mature and form mature photoreceptors. So this kind of proof of concept is possible. So with that, I will leave you, and this is my team of students who had helped us to explore this field. And I have a long list of clinical collaborators and other collaborators with respect to different aspects of our work. And I thank the funding agencies. Thank you. I'll be happy to take any questions.
Thank you, Dr. Indumathi, for this, fantastic talk and sharing this journey. And, we have also seen that how these 3D models can actually take, the research from bench to the bedside. Again, in interest of time, and, it's my apology, I hate to say this, but we will not be able to take any questions.
Okay.
But I guess she can? Okay, we have, okay, good. So we can have questions. Is there, so there is a question there. Can anyone give her a mic?
I'm Shilpa from Vertis Biolab. Actually, my mom, she has 95% retinal damage. Yes. Is it possible to go for cell therapy when there is 95% damage, retinal damage?
Yeah, so it depends on what 95% you mean, whether it is RP damage or including the photoreceptors.
It is RP damage. The
So-
complete outer layer is rugged in.
So the way the pathology works is, in case of an RP-based pathology, RP cells die first. Because the photoreceptors depend on RP, eventually the photoreceptors also die. So when a patient comes at a stage where your photoreceptors are also gone, mainly the outer nuclear layer, which I say, right? So in many of the patients, by the time they can come to the clinic, ONL is totally gone, it's flat.
Yeah.
Okay, in that case, the current therapy, which is RP cell therapy, that many trials are already going on, that's not going to work on those patients because you're putting only RP cells,
Okay.
but not the photoreceptors, because you need both these cell types for a retina to be normally functioning, right?
Okay.
So in these trials, they carefully choose the patients where there are at least some layer of photoreceptors are still preserved, and you transplant the RP cells with an expectation that these surviving RP cells will support the residual photoreceptors to survive and function. So that's the thing. So the clinician makes the decision as to which patient would benefit depending on the retinal condition.
Condition. Okay. Thank you.
Thank you. We can take Devashri.
Yeah, ma'am, I have a question. That was a really amazing presentation. In the retinal organoids, why do you in the differentiation procedure, do you induce epithelial cell polarity, or does it happen naturally in iPSCs?
So we do not induce any polarity by ourselves because these cells, so this is, I assume that you are asking for the RP cells, right?
Yes.
So RP cells, we enrich them at the precursor stage. There's a method of enrichment where they're still proliferating. These are postmitotic, so mature cells are postmitotic. So you may prepare enriched populations and then you allow it, you seed it on a suitable matrix, allow them to mature, and maturation process involves postmitotic arrest, expression of mature cell markers, your polarity establishment because you have ciliary structure, your microvilli projections that keeps coming on the apical surface. All of that happens as the cells gradually mature.
Okay.
Okay.
Thank you.
So we really don't induce any chemically to allow them to polarize.
We can take one last question, maybe.
Excuse me, ma'am.
I think there is one. Okay. We can take two. Fine. You just continue after that. There we'll take.
I have just
Yeah.
a small question. So you said the corneal sheet, which is formed in the organoid cultures, are not as same dimension as the humans, right? It's smaller than the human
Yes.
cells. I mean the human cornea. Can you point out some things like which are the limiting reagents which is not giving the everything to the cornea in the organoids to form the same length? Or why is it shortened if you are giving everything to the organoids?
I don't know, that's going to be really challenging because yeah, we are talking about retina here, or for a cornea which is just, I mean the organoid is just 1/6. What about liver? Liver is a huge organ. How can we make the whole organ structure in vitro? So that's going to be definitely a challenge. Organoids are going to be miniature because of the limitation of your culture vessel. You can do 3D printing for enabling better assemblies because you're here in this case of self-assembly, you're expecting the cells to self-assemble, right? If you don't provide the right condition, the self-assembly may not happen normally. So that gives the variability. But if you are bioprinting, like if you're allowing making pure population of different cells and printing them and forcing proper assembly, then it's possible. But you're not going to mimic, probably replicate the whole organ formation in the dish because of the limitation of in vitro culture conditions. I think that's what I feel, but probably, yeah.
We will take the last question at the middle from the gentleman. Yeah.
I will be really fast. So when you were talking about the transplant, right, in the cornea, you say that when people have been done, this is a success around 76%, something like that. And I was wondering what is exactly means success? Is it the functional is recovered, or is just that you introduce the cell?
If I mean for understanding, so you are asking why only 75% success?
No, actually, I was want to ask what means success. Like, is it the recovery of the function?
What means success? Success is measured clinically So when a graft goes to the clinic, you always do certain QCs with respect to whether these cells express the markers, whether they cover, you have the right kind of graft in terms of, there are different QCs that you do. But clinically, what the clinician looks at is whether the corneal surface is patent, like it remains quiet for a longer time. The epithelium gets regenerated, like you transplant as a monolayer sheet. And it stratifies in vivo, like within 1 week to 10 days, you will see a stratified epithelium developing in vivo. And your vasculatures remain blocked because in LSCD condition, you have conjunctiva coming in, and all of this will become opaque, surface will become opaque. The moment your graft is surviving and stable, the patient will retain that boundary, and the conjunctiva will stop there and will remain there for longer time. And also the clarity, how the cornea maintains the clarity, how much light enters. There are different ways of clinical assessment for that. So based on that, they define a success versus failure. And we see that, yeah, 70% success, that is pretty much good, but then there are also questions why this does not work in remaining 25% of the patients, right? So it's possible that we don't know it, but then it's possible that even if you're transplanting only a sheet of epithelium, your niche, which is there at the limbal stem cell, if it is deeply damaged, and it will not eventually support these cells in the long run, and these patients will not even retain the graft for a longer time. We don't know that, so that need to be evaluated further, probably in more detail in animal models than in humans.
Thank you, Indumathi, for answering all the questions.
Thank you
and,
all.
You know, you can still find her if you have more questions. So let us thank both the speakers, you know, once again, and we will move to the next session.
For chairing this session. We will now break for lunch and meet again at 1:45. Lunch is arranged in the CCMB canteen, which is across the street. It's on the first floor in a hall called Samvad. You can take your coupons, which are in an envelope inside your kit, and you can head there. You could also leave your bags here. We're making sure that the room is secure, so you need not carry everything with you. See you back again.